Where's Nick? Come and say hello to Stan. All right. Have you ever met Stan?
No, I haven't. Where's the camera?
Yeah, there. This is Nick Lohman.
Hi, Stan.
Up and coming, well, already established bioinformatician in the field of microbial genomics. Microbial.
If I look on here,
well, sitting in my chair.
oh, yeah, that's right. Well, very nice to meet you. Thanks for doing this. Is it very early in the morning now?
Yeah.
Okay, just about.
I'm going to teach later today.
Yeah, well,
yeah, it is.
Save you a long plane journey as well.
Yeah, I don't do that.
All right, well, good. You want that more
Well, we're just warming up now. We've done the sound test. You see,
He's had a look around.
He's had a look around virtually. You see his quote and his name on the wall. Next to Charles Darwin.
Very good.
I notice I am absolutely the shortest coat up there. You say that waist and space.
Well, you're so
my other quotes are not.
Well,
yeah. The one about shit is probably one that's not one to face it every morning.
Yeah, I took the point.
hopefully it'll be all right.
Yeah, epigrammatic. That's it. What they call it? The brevity is the art of wit. You've got the shortest quote. Whereas Darwin, Randall's great-great-grandfather, he waffles on. And Carl Rose is actually a bit garrulous as well. Oh, he's got it.
Oh,
Easily, yeah.
All right, well, you enjoy
I will. That's fine.
You can see me and then
I can see. Yeah. So, Mark, I made chapter. I'm going to have to read a little bit because I had a stroke about a year ago.
Oh, okay.
And I'm a little hesitant when I just speak spontaneously now. But the funny thing is, I can actually I can read, and I can read. I'm not going to hum, but it's funny. I can do this better reading and humming than I can just speaking spontaneously. So I'm going to read a little bit. So. I don't want to do it, but I usually don't. I make it as I go ordinarily, but
Well, I might mention just one or two.
I actually just, I miss the spontaneity because I have the word, but I can't.
Well, I have that, and I'm what 30 odd years younger than you. So,
Yeah.
sometimes I just can't remember someone today. Word doesn't come.
Well, I've been teaching, and I wanted to teach the old way that I did, so I would have 100 slides and I'd show in 50 minutes. I can't do that.
I remember you to show some of my slides. You're a bit cheeky, weren't you? I saw one of your talks and you borrowed one of my slides.
I didn't borrow it, I played.
Yeah, you played exactly. That's it. Great artists don't steal. They don't great artists don't just steal. Yeah, that's it. That was it. Picasso said that.
And I did it probably without attribution. Yes.
That's true. Exactly. So what actually happened was that a few years ago we were setting up a new model, and I thought, well, now in the modern age, students don't have to rely on my lectures alone. I ought to give them a sampling of what's out there on the same subject elsewhere. Let's have a look. And I found your lecture that was recorded. And I thought, oh, just better check it's okay. I went through it. And I got to about 20 minutes, thought, hang on, that's one of my slides. I'm making that on my Mac with Barts in the early 1990s. He stole that attribution. I went on another 20 minutes and found another one. And then I was thinking, well, I can record my lectures and put them up on YouTube. But people will say, have you made sure that every last image you have an attribution or you have copyright to do it and all that kind of stuff? And that's what the university's official line is, that you can't do anything unless you've checked out every last piece of copy. I thought, if it's all right for Stan Falco to shove stuff up without any of this worry, I don't care. And so since then, I've recorded everything I've done. I've got about 30 or 40 lectures up there on YouTube with all sorts of images taken out of journals and whatever. And nobody has come to me and said, you bastard, you plagiarised me, you owe me £20,000 or... Actually,
you have fair use.
Well, in the US, you can, yeah.
Yeah. So that's fair use.
Yeah, exactly.
But not without attribution. But I'm old and people forget me. So.
Yeah. I'm slightly old, I'm faster than you, and I've got a bigger mouth. So, strange waiting. How many more have we got to come? There's still a lot. Yeah, just waiting for them.
You're early.
We are early. We should have some news saying or something. Yes. Yeah. Well, John could do a warm-up. Barbara Brinkman, when he used to do his stuff, he used to pace around outside before he did the show. He used to sort of try and get himself worked up into the thing.
Well, John's herbal tea.
That's it. No, it's good. I'm not suggesting you go for the chair.
No, I'm currently trying to encode all this information into my brain.
Okay. And turn it into a freestyle. Yeah, well, I suppose we ought to do the AV over here. So how is this working? Is it working for you?
Yeah, it's working.
And where are we streaming to? To the
White Medical School.
White Medical School. So have you tweeted that? Check it out on White Medical. What is it? Someone find the YouTube.
Yeah.
Everyone retweet Jazz's. Retweet it so that everyone finds it. Because there's people out there all across the world waiting for this event. But there's still people somewhere out there. Yeah, exactly. Yeah.
Have you got some when you wipe the hotspots?
Yeah. Yeah. So it's when I set up Skype the other day, we kind of thought that it wasn't that great. And so I've wired it just to make something that much better. And this is pretty good, isn't it? Press it on. It is,
And I'm going to do that.
Stan just says, you know,
yeah, I know. So I just want to begin I'm into funding science as well. I still do that.
looking forward to the
Yeah, it's just so much to do.
So we you're so annoyed that you can transfer across the so annoyed about that. I want my best shirt. Yeah, I don't know where I'm at.
Brilliant, yeah, and then there won't be any that's fantastic. And you can use your
Chris, come in. This is Chris Quint. You met him? You heard of him? He's a cool guy. It's like I've got around waiting with that. He was in on our JAMA paper and full part the German essay. You want to save the paper. Well, he had his so it's David London. We're almost certain, David almost confessed, was the expert that the editor of JAMA was consulting to see whether our paper was going to pass muster. And he said, you've got to solve this whole thing de novo. You can't just go and lie against the records you know that you're having that up. And that was a hard ask. Nick, how long did you have? You have to do that about 3 days. 3 days?
Well, what, to do our assemblies?
To do the whole thing de novo.
Well, yeah, about 10 days.
Yeah, exactly. And Chris came in and said, good. You worked in there. I really worked. Half an hour's inspiration, 2 years plus. It's just got to. We'll be ready in 5 minutes, okay?
Okay, fine, yeah.
We've got a group of people who are teaching at the moment. The medical school does this case-based learning. And they've been teaching case-based learning. They're coming any minute now, totally bagged out for the day working with students. Oh, they're here. Oh, you are here.
Well, actually, that's what I'm going to.
Is it? You're doing case-based learning?
I'm going to go to the case-based learning.
Oh, there we are. So they can feel sympathy with you, and you can feel sympathy with them. I've managed to dodge the bullet on that one up till now. But I treat Mark as a role model. If you just become a radical, I'm not teaching. Randall? Yeah. Because elements to yeah, and I always yeah, it's very professional. It's a lot nicer now that the finished all the work. Yeah. Yeah. You came over by train? No, I doesn't say. How long did it take? Took me about the same way I came from Cambridge. I really got stuck on the M1 maintenance. Isabel, you're in trouble. Like Jane.
I did.
She says you didn't.
I did.
You must have said the wrong Jane. No, I don't know. Anyway, she's here now. Everyone's here. Yeah, that's it.
Oh, yeah.
Now, what we're doing with the lights as well. There's a way in which you can turn big lights on. Put these just side lights. These things. So come here. Sam, this is Randall. Hello.
Hi, Randall.
He's come up from London to give us a little talk about his great-great-grandfather's ideas about the tree metaphor and evolutionary trees.
How can you, are you seeing me straight?
The top of your head is a little cut off.
I see. So,
You're not going to leave me up here the whole time. You're not going to leave me up here the whole time.
No, we're not going to leave you up there the whole time. No. I will turn you off if you like. Are you feeling?
I mean, I think people are going to get very sick of me, and if I went and scratch my privates, I can't do it.
We'll turn you off after you've spoken in about half an hour. Is that
That's fair enough.
Or we'll take off the screen, yeah?
Yes, I can
This
disappear.
is Stan Falco. This is great Stan Falco. But he doesn't realise how big he appears to us. This is a fitting tribute to his dominance of the intellectual landscape as well. There we go. Anyway, it's the time, isn't it? Is there anyone that we know is not here? Yeah, always Mark. Mark, you have to sit at the front because you're late. Come and sit at the front, come on. Just like in lectures, nobody wants to sit in the front. So everyone in the background, move to the front row. Come on. Come on. Nick Logan, come on. Oh, you can stay because you're going to live tweet. Yeah. Come on, come at the front. No, it's more comfortable. All right, then, well, we're going to get started. So, thank you all for coming. Just over 2 years ago, we made a tryst with destiny. It's now time to repay that pledge. How did that come about? Well, just over 2 years ago, I was working out my notice in Birmingham, and I got an email from David Rand, who said he would be here, but he isn't here. There we are. And he said, well, the MRC are just launching this initiative where they want to put money into bioinformatics infrastructure. And he said he was a bit too busy. Could someone else take it on? And I thought, oh, I don't even work there yet. But one of the things that was ringing in my ears from the interview was that David Rand had said in the interview when I said, oh, I've got a couple of project grants, and he sputtered about project grants aren't enough. And so I thought, well, there were pressures on, I better do something. But having, the thing was, the deadline was the 22nd of April, so just over 3 weeks after my start date here. I thought, well, I haven't got time to get to know everyone, work out who works on MRC stuff and who might be a contributor. And I did what the advice they give to authors is always write about what you know. So I thought, well, I can sling something together on medical microbial bioinformatics. And I've just been working with Nick until recently, and I thought it would be good to work with Nick again, particularly as we'd had a bit of a tiff, and I thought we need to make up. So I thought, well, we'll get Nick in as well. So I thought, we didn't quite kiss, no. My daughter does claim that we have a bromance. There we go. As a result, I slung in something within the deadline from us in Birmingham. And thought, well, we'll see what we get. We've got a distinctive remit. You just missed me quoting Nehru, only mirror it. And you didn't catch the illusion. Shocking. Anyway, yes, so where was I? I was saying that, yes, so we put that in. We got a response from the MRC saying, yes, you can go through to the next round. So we're very pleased with that. But they said, there is this catch. And I said, what's that then? Well, you have to work with this guy from Cardiff and this guy from Swansea. To which Nick and myself spluttered with indignation. And there's this thing called the Haldane principle, which says that government doesn't set what scientists do, scientists say it. And I thought, this is like the Haldane principle gone wrong. The MRC can't tell us who we have to work with. I mean, if they're telling us to work with someone from Oxford and Cambridge, maybe we'd listen. But not Swansea. But anyway, we bit our tongues and ran off with it. And it turned out the guy from Cardiff actually was from Cambridge, and the guy from Swansea actually was from Oxford. So that went okay. And not only did they turn out to be extremely intelligent and capable microbiologists working at the edge of bioinformatics and genomics and pathogenesis, but they also turned out to be two highly agreeable individuals who we had no trouble working with at all. And one of them in particular, who's got his arms crossed over his large girth, brought in an additional ingredient. So I had what you might call the gift of the Blarney. I could actually write a load of bullshit to persuade the MRC to give us money. But the amount of money on the table was quite substantial, and it was heavily biased. They said it has to be spent on capital, on infrastructure, on computers, on buildings, not on people. And I was saying, well, can we really justify asking for millions and millions of pounds worth of computer equipment just for medical microbial bioinformatics? And Tom just said, yeah, of course. The rest of the world, the rest of the country should have exactly the same capability as the same. Why should we be poor cousins? Just ask for millions. We ended up asking for 3.7 million pounds worth of computers spread among 4 universities. We asked for £700,000 to do this space. We asked for over £1 million to refurbish space in Swansea. So that essential ingredient, some of our Jewish collaborators and colleagues will recognise the word chutzpah, and that's what Tom brought in. We just did it. We just asked for millions of pounds, and we got it. It turned out that we got the money, got word that we got the money just at the end of that year, 2013. And again, there's always a catch. The catch this time was you can have all this money, but you've got to do all the capital spent in the next financial year. So you've got to spend all this money between April the 1st of 2014 and March the 31st in 2015. So we did it, basically. That's what we're here to celebrate. We actually managed to spend £3.7 million for computers, £700,000 in here, over £1 million in Swansea. In addition to doing that, we recruited 3 very smart fellows into fellowships. We recruited excellent support staff, Isabel and Ben, who've done a fantastic job, and basically we wouldn't be here if it weren't for them continually pushing the process forward. So any of you who've not worked in the university sector for very long will, may not realise actually getting anything through finance departments and all that kind of stuff is not easy. So actually forcing this thing through, actually finance is easy, estates even worse. But luckily we had that deadline and we had to get the money spent, otherwise we had to give it back, and we managed to do it. So there's one more thing that I have to mention, which is that it was not just the spending of the money, it was the building that had to be done. And a lot of people were very inconvenienced by the building, the noise, and just this area was just a building site for well over 6 months, and the people in the offices nearby, some of them who work with us now, people in other offices nearby and downstairs and whatever, all inconvenienced. We can now celebrate today that that is over, and we now are taking ownership of this space, and we're going to start using it from now on. So that's basically congratulating us on getting this far. Obviously, with any grant and any project, actually getting the money and getting the wherewithal to do it is only the first part. We still have a challenge ahead of us, and that's a big challenge where we are actually going to have to get the infrastructure in place, the hardware working. It seems to have been plugged in, the acceptance testing seems to suggest it works. We're now going to build the layers of software to actually get us to the stage where we can offer what we are calling cloud computing to the whole of the UK medical microbial community and beyond. Hopefully we'll have international partners and we'll have partners from outside the academic sector. And that's the challenge that's rolling out in front of us now. So our struggle's not over. It's a bit like Nelson Mandela said at the end of his book, you know, once you've climbed one mountain, you see you've just got another one to climb in the distance. But at least we can pause now and celebrate. So that's all the kind of oratory out of the way. The main thing I've got to do now is introduce three remarkable people who've given up their time to come and join with us and help us celebrate this event. First of all, and I'm going to introduce them all in one go, even though they're going to speak separately, so that we go through a seamless transition. And I think at this stage I might get my notes out, just because some of them are so eminent you can't remember everything about them. So Stan Falco is here, joining us from Stanford. He's now 81 years old, so he makes some of us around here look quite young. It certainly makes me feel quite young. Stan is really one of the well, the godfathers, is that the right word? He's the forefathers, the founders of our discipline of molecular microbiology. He, as an undergraduate, was at Maine, then he did his PhD at Brown, and he really got into his stride in the 1960s when he started working on pathogens like Shigella and showing that they had these things called plasmids, these bits of DNA that work on chromosomes. And he showed, after that, he started working on E. coli, developed a abiding interest in diarrheogenic pathogens, showing that those plasmids were involved in antibiotic resistance and also in pathogenesis. He was actually there at the birth of molecular biology, rubbing shoulders with Boyer and Cohen on one of their famous papers. They acknowledged they got the strain they used from one S. Falco. So if that's not enough, well, he also then developed interests in things like Neisseria and Gonococcus and H. pylori. And in fact, he's worked on so many different organisms, and he's actually justified his choice of working on so many different organisms by this epigram that we've put on the wall here, which is, he said, I've never met a microbe that I didn't like. And it gives me great succor to think that you can actually work on such a broad canvas. Some people tell you, oh, you must always just work on one organism, become a world expert on one organism. And when you think about people like Stan, it's clear that you don't have to do that. For those of you interested in, like, you know, the size of a man's H index as a measure of a Stan's H index, according to Google Scholar, is 138. Okay, so that makes the rest of us look like dwarfs, doesn't it? Stan's also had lots of interest in the interface between bacteria and the host. He's interested, key player in vaccinology. He's very widely quoted for his work on what are now sometimes called Falco's postulates, otherwise known as molecular Cox postulates, which he put forward in the late 1980s to try and bring some clarity on how you work out whether a gene is involved in virulence. And he said you have to knock it out, show that the virulence goes away, put it back in, show it comes back again, and then investigate in other ways. What else is there to say? Well, he's won plenty of awards. He's a fellow of the Royal Society, one of the few that have been given given out to people from outside the UK. He got the Lasker Prize, which is an American kind of equivalent of Nobel. But I think also it's worth stressing that he's almost set up a dynasty or a tradition. So many key workers in our field are just 1 or 2 handshakes away from Stan Falco. So this man here, for example, is 1 handshake away. He worked with Stan for 6 months. You told me, when was it? 1978. And the one abiding memory he has of Stan was that Stan told him that you must go and handle Vibrio cholerae in a safety cabinet, because he was working in Seattle in a port, and if the Vibrio cholerae got out of the lab, then the port would have to be closed. That advice has stuck with me for all these years. My supervisor for my PhD, Gordon Dugan, has worked with Stan. David Relman, who you may know as a prominent worker in the field of bacterial pathogenomics, has worked with Brett Finley, Rena Rapparoli. All of these people have worked with Stan. And some people have met up with Stan in other situations as well. So Jared Diamond, who you might have read some of his books, actually is an associate of Stan in his leisure life, working in the Bitterroot Valley in Montana, fishing together. And in fact, in one of Jared's books, he opens a chapter talking about his experiences fishing with Stan. Anyway, that's enough about Stan. I'm just going to, I said I'll go through these 3 quickly, so that we then can go on seamlessly and let them talk rather than me talk. In addition to Stan, we have Randall Kames, who's come from London, from Islington, where he lives, to join us. Randall has a very famous pedigree in the traditional sense of having some extremely famous forebears. So some of you will know that his great-great-grandfather happened to be Charles Darwin. You might not know that he's also descended from related to David Hume. David Hume didn't have any children, but he descended from David Hume's brother. His great uncle, you may have heard of as Maynard Keynes, the famous economist, whose influence perhaps should be coming more to the fore instead of displaying austerity around these days. His grandfather on his mother's side was Lord Adrian, who won a Nobel Prize for his work on nerve cells production. His son actually has taken on a little sideline of some note, in that he starred as a film star, as Edmund, in The Lion, the Witch and the Wardrobe films. Anyway, with all that on his shoulders, it's no surprise that when he was a young man, he rebelled. The family said he had to go to Cambridge and study science. And he said, sod that, he's such a rebel. He went to Oxford and he studied Anglo-Saxon and other slang. It was a way of turning his nose up at the family tradition. After that, he then found his way into the civil service and worked in the Ministry of Defence. And some of you know, I've just come back from Northern Ireland on holiday. He was in Northern Ireland, but he was there at a time when things were a lot more troubled than they are now. In fact, he used to spy on Gerry Adams in his former life. He has, throughout his life, developed an abiding interest in the legacy of Charles Darwin, and he's currently involved in various projects, trying to get Down House, maintain Down House, get down to Darwin's residence, getting UNESCO status. He's worked in the Galapagos as well, working there to ensure the legacy of Charles Darwin. He's still in the Charles Darwin Trust,
yes.
Working with young people to use the legacy of Darwin there to energize the arts and poetry. And he's also has a particular interest in working in the Arab world, because his wife is Lebanese, and has actually been working with the British Council and spreading the message of modernity and the legacy of Darwin and rationalism in that setting. Finally, we have John Pace, who's joined us back there, who has come to do some rap music for us. John did a BSc in science and science fiction, and then a BEng in aerospace technology, before deciding that actually science communication was his thing, and he did an MSc at the University of Glamorgan. Since then, after a short time working directly for the University of Glamorgan, he's been working as a science communicator and has had all sorts of roles, working with CBBC and CBBS, working with Open University, Bite Size, doing as much as he can to make science accessible and exciting, particularly for young people, and particularly through the medium of rap music. He is also a very keen astronomer and interested in the evolution of the universe and then the evolution of life and things like astrobiology. And one of his stage names is Kurt Kuiper. And those of you who know a bit of astronomy will recognize that, you know, these are regions of the solar system far, far on the limits of what we can see. And he's very excited at the moment because the New Horizons probe will be showing us Pluto in a few weeks' time in the Kuiper, a major Kuiper belt object. Anyway, that's the introduction. I'm now going to pass over to Stan, who's going to say a few words of encouragement and remind us that these are the good old days.
I am delighted to join you here today, and I'm a bit humbled by Mark's introduction to me. An awful lot depends on who gets born first, and I had the good fortune before when things were easier. So I want to add my congratulations to the chorus of people who've come forward today to celebrate the opening of CLIMB, which I understand is the cloud of infrastructure of microbial bioinformatics at work, and it's an admirable enterprise. It's a consortium, as you know, of Birmingham, Cardiff, and Swansea, as I understand it, and for the public and private sector in all of the UK, and to help understand bioinformatics of microbial life in all aspects. I really envy enormously. You are the generation of scientists that bring software and data storage and bioinformatics to be able to track the epidemics of the past and to study in great detail the transmission and the dynamics of infectious diseases. And it's a great privilege that you have. I hope that you appreciate it. And I hope the people of the UK appreciate the reference. You have the ability to examine both the genome of the offending microbe as well as the host, including us, in addition to my support. So I'm reminded, I can see Mark Ockman sitting prominently in the front row. So I'm reminded actually at this gathering of people that at the time in 1978, when he and Gordon Nugent both were in my laboratory at the University of Washington, and we were at that time able to sequence 75 base pairs a day, we would analyze our results on a Radio Shack TRS-80 computer with 16K of RAM using a program that I wrote in BASIC. So I think we've come a long way in 37 years, but I still remember the thrill of my first ATG codon in the sequence of the E. coli heat label enterotoxin to this day, actually. So I can only dream about what you're going to accomplish in the next decade. I wish I could be there. Maybe I will. But I'd enjoy their accomplishments, and I may say, I don't think that you can even dream of the accomplishments you're going to make in the next decade. You're in for great surprises. The good old days are now, I remind you, and it's worth saying. So I wish you good luck and Godspeed in the adventure that awaits you. It's a privilege really to be here and join you in this celebration.
Now, Stan didn't want to necessarily be on the screen all the time now. You can take him off.
Let me see the slides.
Yeah, so you can still see. He's not using slides. He's just going to talk from the cup.
Okay, that's fine. I can see.
There you go.
Thank you so much, Mark, for the invitation to join you here this afternoon. I was excited when you explained what this was all about, and I'm absolutely thrilled, especially by what Stan has just explained to us. I was sort of sensing that with my layman's understanding of this whole field, and to hear his account from his position, and to see you and to think about opportunities and so on is just remarkable. I'm really here today because when Mark told me about Climb, I was fascinated and impressed by the boldness of the plan. Managing all these fresh kinds of information with cloud power and flexibility for all the intriguing investigations you are now working out how to tackle. Mark suggested I might say some things for Darwin, and I felt at once, yes, if Darwin could be here with us today, he would be fascinated and impressed. With you, there can be no doubt about that. I think, excited. He'd see the opportunity clearly that Climb presents, and he would sense the spirit of the whole venture. And I caught that just this afternoon, meeting Tom and the others of you. And it's very strong to an outsider. You may be getting used to it, but it's strong. Darwin would be eager to hear about every plan and delighted to talk. I thought if it would mean anything for you to know my feeling about that point, I'd be glad to tell you for his sake as well as yours, because this venture is one of so many continuations of his initiative with the Origin of Species. And I feel somehow that all who join in the effort in these ways are joined with each other in it. So you're with Darwin, we're with Darwin, he's with us. And that's the nature of the whole activity that we're involved in. So let's just keep Darwin in mind for an understanding with him of how far we've come from his work to here. And in the same way, with Koch, with Cohn, and with Vosa, if I pronounce Vosa right? W-O-S-E. Wow. Yes, sorry, yes. Each of them had some luck. Now that luck was their opportunity. They seized theirs. You all had your opportunity with Climb and are seizing it. That's what this is all about. And please use it boldly where and as you can. What I'd like to do, first of all, is to just roll those words round again. Cloud infrastructure for Cloud infrastructure for microbial bioinformatics. Wonderful, wonderful. I, with my background in work with information, find the bioinformatics especially interesting because I have my own understanding in other areas of the huge gains to be made by managing information carefully, imaginatively, and really boldly and competently. And I would reckon, just with bioinformatics, you're really going to have doors opening and advantages to make. There are wonderful words for an onlooker like me. Let me now just touch on some points that I feel link you and what you're doing with Darwin. First of all, the evolutionary emphasis. Mark took as his text for his inaugural lecture Dobzhansky's insistence that nothing in biology makes sense except in the light of evolution. We know that well. Mark showed how true that is for microbiology, closening the look. And he's explaining to me today, closening the focus again, how central that is for so much of microbial bioinformatics. Now, there's one key point, it seems to me, in this, which is, and this is as a layman talking, and this may be very banal, but it's the speed of reproduction in microbiology, with the extensive variation, the selection of the process, and the significance of the changes for other organisms. It's a truism how difficult we find it to observe evolution, actual evolution of macroorganisms. There are the few well-known examples, but for all other organisms, it's like grandmother's footsteps, with the stillness whenever you look behind you for any movement. But microbial genomics can be, as I am coming to understand, a helter-skelter ride. In this area, excitingly, the often bucking process of change is a key concern. It's a central issue that we must understand. On from that to the tree of life. Now, Darwin wrote in The Origin that relationships between all species of the same kinds, as he said, have sometimes been represented by a great tree. That's the tree of life, no less. He wrote carefully, I believe this simile largely speaks the truth. And then he wrote, the green and budding twigs may represent existing species, and those produced during every former year may represent the long succession of extinct species. And in this way, the great tree of life, in his words, fills with its dead and broken branches the crust of the earth, and covers the surface above with its ever-branching and beautiful ramifications. Now, it's important to recognize here that the essential point in all of this for Darwin, when he wrote, and for Klein, is not surely the huge trunk, but those ever-branching and beautiful ramifications. This is actually Darwin's endless forms, that last sentence of The Origin of Species, in which he wrote about endless forms, most beautiful and most wonderful, and how they have been and are being evolved, are being. Those three last words of The Origin of Species, because that's what they are, about the ever-continuing process, are, as I understand it, central to what Klein is looking at and about. On from that quickly to big data. I first heard this watching Tom's presentation on YouTube, I think, of the Klein project, which was very helpful, wonderfully and pointed and helpful. So we have 4 petabytes of data storage and 78 terabytes of total RAM. Is that? That's right, yes. That is a lot. Yes. The need for such capacity follows directly from the understanding of evolution that has stemmed from Darwin's writing. With its randomness and variation, I think those are words of yours that I've picked up, its endless proliferations, that's one that I focus on, and the whole global diversity of life, in which microorganisms indeed excel even macroorganisms. Hearing talk of big data, I think at once of the number of data mountains that Darwin had had to climb through his working life to gain an adequate understanding of the topics he was having to tackle then. Especially, he would have said, the 8 years he had to spend dissecting barnacles in order to prove himself as a competent taxonomist, so that he could write as he wanted to on species as a general topic, any or all around the globe, and gain any serious attention for his views. For the 8 years' time that the barnacles took Darwin, remember his poor son, who was then staying with a friend at the age of 10, looking around the house, and he asked, "Where does your father do his barnacles?" For Darwin's understanding of the endless variety of natural life, the infinite complexity of all the interactions it involves, he would appreciate at once why Klein is focused so sharply on the scale of data storage and processing capacity it's aiming to provide. On to medical vaccine. When Mark Pallen first explained the project to me, I saw at once the great interest for pure science. But when he explained about some of the investigations for which it is to be used and the MRC funding, only then did I really take in its significance for medicine. Picking up its potential for work on hospital infections and antimicrobial resistance, I remembered a point about Darwin. Now, not about an achievement of his, but about his reaction to another person's achievement. How quickly he recognized it, how strongly he felt about it, and this is something about the science and about its value for medical treatment. So this is the point. First of all, I have to explain that when Darwin was working on the origin, his eldest daughter, then 10 years old, called Annie, fell ill. Probably, we've now worked out, with TB. He was devoted to her. He did all he could to save her life, caring for her night and day in her last illness, and taking notes through the night, and then the next morning, the treatment he gave her, her temperature, how she was during the day, and so on. He was watching her, anything he could do. And he was devastated by her loss. He was deeply shaken by the physician's inability to help. 25 years later, in 1877, as Pasteur was making the case for the germ theory of infection, a close scientific friend of Darwin's, Professor Ferdinand Cohn, now is he well known?
Yes.
Yes. He was then a plant physiologist, but he later became the founding figure of microbial taxonomy. Cohn sent his friend Darwin a copy of his periodical on plant physiology. But this issue contained the first photographs ever published of bacteria. Anthrax bacilli. They'd been taken by Robert Koch, who was to identify the tuberculosis bacillus 5 years later. Koch had come to Cohn with his first microscopic preparations of the bacilli responsible for anthrax, and had shown him his paper, arguing for the first time that these bacilli were the cause of the disease. Professor Cohn had recognized at once the great importance of his findings for medicine and the preservation of human life, and commented to Darwin in his letter that the photographs showed the least but also perhaps the mightiest living beings. Darwin replied, this is the point, "I well remember saying to myself between 20 and 30 years ago, that is, while round about the time when Annie had died, that if ever the origin of any infectious disease could be proved, it would be the greatest triumph for science. Now I rejoice to have seen the triumph. Now that was what Koch's success with the anthrax bacillus meant at once to Darwin, the scientist, and the farmer. Now for information management. I was fascinated to hear Tom Connor's explanation of CLIME on YouTube with his picturing of the sequencing iceberg and his breakdown of the costs of different parts of the project. The 75% for bioinformatics expertise was a critical need for the project. It was a bottleneck, I think, he said. And it's good for me to see this need recognized. I went into the civil service after university and I worked on different sets of information, kinds of information in my working life. Often large amounts of very poor, corrupt, incomplete information, trying to find ways of getting what we could wring out of them for important needs for understanding, knowledge, for action, and so on. It's so good, I'll say first, to see how CLIME users will be drawing in or reaching out to other data sets besides their own, combining other kinds of information with their genomic data, with data sets with different textures. These will be obviously clinical, diagnostic, population, epidemiological, and so on and so forth. Now, as I say, I've worked in government and the NHS for a while, often in areas needing this careful and effective management of huge quantities of inconsistent and unreliable information. But so often, the information management needs have just not been recognized by management and they've not been provided for. I've found in life that many people who I would expect to understand just don't see the problems. They don't see the effects of the problems as they seem to feel information is just information, simple. And they don't see a need for ordering and control and so on. People are often perfectly good with their own data simply because they know it well, but are then casual and careless about others. And when they use their information alongside others, they don't see the potholes until they find themselves stuck in one. You are well-resourced, well-staffed for that work. You're focusing on those needs for management of the kinds of information you're putting together, the disciplines you need, also, I imagine, the creativity with flexibilities of different kinds, so that you can try combinations of this and that. And from all my experience, that sounds really good. And I would very much hope that you'll be making remarkable gains in that kind of use of information in the coming years. I've been hearing about antimicrobial resistance, the review and the value of CLIME for that work. When you join and work between different kinds of information, you have a challenge. Now, I was struck by a phrase of Mark's again. In one of your lectures, I think, you toss out a phrase, night's move thinking. Do you remember that one?
Oh, I don't remember that one. I was showing it at the time.
Very good. Well, what it means, it's another version of thinking round corners. And I'm sure that your CLIME is going to greatly ease your ability to think round corners, make night's moves that suddenly get you able to deal with something in a fresh way, combining different things, and go for it. I'll just conclude, that's enough about Darwin. I'd say with the whole approach, be bold. You've got, I don't need to say that obviously, because you're all very bold people and you're thinking big, you're thinking, and you've got all your ambitions. What I've been hearing about the projects, the range of projects that you're now working on, and sort of eager to apply all the power to, it's extraordinary. Now, if Darwin couldn't offer you anything, I would like to suggest that you just remember a phrase of his, which he uses in one of his most private notebooks, the one in which he came out with the branching diagram, I think, and then the branching diagram. In another point, he writes that he should let conjecture run wild. Let conjecture run wild. Just bear that in mind, and if ever you can see a possibility for an exciting idea, follow it. He had in his life, he explained once to his son, he had a phrase, fool's experiments. And fool's experiments were ones that he would try for himself in his mind and not tell anyone else about, because he expected that they would laugh at them. And he found that they were valuable. Be bold, let conjecture run wild, fool's experiments, just use everything you have. And the final point from Darwin, again, for you here now with the climb opportunities. Among almost the last thing he wrote for publication before he died was a preface to a brilliant book by a friend of his about adaptations of flower forms for cross-fertilization by different insects. They were looking at all the range of flower forms that they'd looked at and worked out the functions of for cross-fertilization and seeing extraordinary things. He ran through 2 pages of things that he had found in the book and would like to work on himself. At that time, he was failing. He was pained with angina. He could no longer stand at his microscope. He knew he was dying, but his mind was racing onwards. He, though, simply wrote, it is superfluous for me to make any further suggestions. These will occur in abundance to any young and ardent observer who will study Professor Muller's data and then, and this is it, observe for himself, giving full play to his imagination, but rigorously checking it by testing each notion experimentally and analytically. I'm sure I'm adding that last word. If he will act in this manner, he will, if I may judge by my own experience, this is Darwin talking, receive so much pleasure from his work, and we can add success. So that's it. These will occur in abundance to any young and ardent observers. You are ardent observers. You have the tool, and over to you.
We were going to get this cut of ribbon, but we couldn't think of any easy ways to do that. Yeah, we're going to get you to uncork this champagne. Health and safety dictates you must point towards this corner, not towards the screen, not towards the audience. So, this corner, I say before I open. Yes. I don't want the screen to be destroyed.
So here we are. Yes.
We'll do a symbolic test.
Who for the second time?
Well, you and I do one quick and then everyone will drink.
Yes.
Interlude. So I'll wrap up.
Yes. Let's see.
Cheers. At your age, you could drink champagne for breakfast, surely. Churchill won the war on champagne, you know, that's what he drank. Good, okay, so Isabel, come on, quick, need champagne, everyone. We're just having a little interlude now, just everyone can relax for a few minutes. There's some refreshments out there, and then in maybe about 10 or 15 minutes, John will set himself up and be ready to do some rap music. Okay, so.